Joe. Welcome to MCAT Biochemistry. This presentation is going to be about introduction to protein structure. This is going to be a pretty long presentation, so please be ready with your notes or just listen to it carefully. All right, so let's start. Let me introduce what proteins are. Proteins are large biomolecules and macromolecules that comprise of one or more long chains of amino acids residues. All right, so they're made of amino acid residues. They may have one or more long chains. Proteins perform vast area of functions within organisms, including catalyzing metabolic reactions, DNA replication, responding to stimuli, providing structure to cells and organisms, and transporting molecules from one location to another. So proteins differ from one another primarily in their sequence of amino acids, which is dictated by nucleotide sequence of their genes, and which usually results in protein folding into a specific 3D structure that determines its activity. So the specific 3D structure determines its activity. Uh, Proteins differ from one another primarily by sequence of amino acids. The sequence are actually dictated by nucleotide sequence of their genes. All right, so genes are the one that decide the sequence. A sequence of amino acids are dependent on protein folding structure into specific 3D structure, and the 3D structure determines the activity, the function of the protein. So remember that the structure of the protein will be defining its function. A linear chain of amino acid residue is called polypeptide, and a protein contains at least one long polypeptide. So, a protein would always contain one long polypeptide. Short polypeptides containing less than 20-30 residues are rarely considered to be proteins and are commonly called as peptides. So, there is basic difference. Anything less than 20 to 30 residues would be called as peptides. Anything longer than that would be a polypeptide. Individual amino acid residues are bonded together by peptide bonds and adjacent amino acid residues. Alright, so individual amino acid residues are bonded together by peptide bonds and adjacent amino acid residues. So, uh, they're usually bonded together by peptide bonds, less than 20-30 amino acid residues and adjacent amino acid residues. The sequence of amino acid residues in protein is defined by sequence of a gene, right? We just discovered that, uh, which is encoded in a genetic code. In general, genetic code specifies 20 standard amino acids. I have another lecture on amino acids, and you're more than welcome to go and check that out. But in certain organisms, genetic code can include selenocysteine. In certain archaea, pyrolysine archaeas are actually organisms that survive in a very difficult situations, let's say hot springs, environment that cannot be survivable by normal living organisms, right? So those are archaea. So archaea may contain one more amino acid, pyrolysine. Shortly after or even during synthesis, the residues in a protein are often chemically modified by post translational modification, which alters the physical and chemical properties, which includes folding, stability, activity, and ultimately the function of the proteins, right? So, shortly after or during synthesis, the residues in protein are often chemically modified. So, the protein actually goes through chemical modification, post translation modification, which may change their physical and chemical properties and how they fold, how stable are they, what kind of function can they have, and activity, what it can do. Some proteins have non-peptide groups attached, which can be called prosthetic groups or cofactors. All right, so some proteins may also have non-peptide groups. Okay, so it may not have 20 to 30 amino acid residues connected to it, right? They may have what you call cofactors or prosthetic groups. Proteins can also work together to achieve a particular function, and they often associate to form stable protein complexes. All right, so teamwork makes the dream work. Proteins usually come together, all right, and they associate to form stable protein complexes. Your stability will also define your function, right? Depends on how stable protein it is. It will be more functionable. So remember that teamwork makes a dream work. Most of the proteins come together to form stable protein complexes. Once formed, proteins only exist for a certain period and are then degraded and recycled by cells machinery through the process of protein turnover, right? So there is something called as protein turnover. So proteins only exist for a certain period, right? So it comes with an expiration date. However, after that it gets degraded, it gets recycled, all right, by the cell's internal machinery through the process of protein turnover. Okay, so understand that there is an internal recycling process within the cell's machinery, which they call it protein turnover. A protein's lifespan is measured in terms of half life and covers a wide range. Okay, so protein's lifespan can be measured, it has a unit, right, of its half life and covers a wide range. They can exist for minutes or years within an average lifespan of 1 to 2 days in mammalian cells. All right, so for all the mammals, they exist for minutes or years, but the average lifespan in all the mammals is usually 1 to 2 days. Abnormal or misfolded proteins are degraded more rapidly, either due to being targeted for destruction or due to being unstable. Okay, so sometimes they are targeted for destruction or they are unstable, and that's why they just get degraded. And they're like, okay, well, we don't think so. You're functioning, you may not be normal, or something's not right, your folding's not right. Let's just degrade you, and we'll have more space of other better functioning proteins, right? So they have a very cutthroat approach to functioning. All right, so like other biological macromolecules such as polysaccharides and nucleic acids, proteins are essential parts of organisms and participate in virtually every process within cells. Okay, like other biological macromolecules such as polysaccharides and nucleic acids, proteins are essential parts of organisms and participate in virtually every process within cells, which is true. Our body is more functionable because of proteins itself, so it's very essential for all organisms. Many proteins are enzymes that catalyze biochemical reaction and are vital to metabolism. Yes, enzymes are very important. They actually act as a catalyst in a lot many functions in our body, right? Proteins also have structural or mechanical functions such as actin, very important, myosin, very important in muscles, and the proteins in the cytoskeleton, which form a system of scaffolding that maintains the cell shape. All right, so we also have a certain proteins that help with scaffolding and maintenance of the cell shape, right? So we have studied actin and myosin, and we have other proteins that help cytoskeletons, right? Other proteins are important in cell signaling, immune responses, cell adhesion, and also the cell cycle, how one cell is connected to another, cell signaling, what is the process of signaling between the cells, inter-signaling between the cells, and proteins play a very important role. Proteins also play a very important role during the cell cycle. The unique sequence of amino acids in polypeptide chain is its primary structure. The linear sequence of amino acids in polypeptide chain are held together by peptide bonds and result in NCC pattern backbone. The primary structure is coded for in the DNA, a process you will learn about in the transcription and translation modules. So I am going to have a module regarding transcription and translation. We are going to learn about primary structure coded for in the DNA, right? So we will be talking about it. Just remember that unique sequence of amino acid in a polypeptide chain is its primary structure. The linear sequence of amino acid in the polypeptide chain are held together by peptide bonds and results in NCC pattern backbone. The primary structure is coded for the DNA, a process that we will learn in transcription and translation. All right, so primary structure is coded for in the DNA. The peptide bond between two amino acids is depicted. The shaded quadrilateral represents planar nature of this bond. So the shaded quadrilateral, it represents the planar nature of this bond. By planar, we mean that two alpha carbons, the nitrogen and the carbon and oxygen associated with the peptide bond, all lie in a single plane. As you can see, they are all lying in single plane, right? So that itself means the two alpha carbons, right? The nitrogen, and the carbon oxygen. All right, so they are all in single plane, that is the planar. So this is alpha carbon 1, alpha carbon 2, that is the peptide bond is a carbon, that's a carbon. We have nitrogen, we have oxygen, neutron, right? So this is all planar. However, each amino acid can twist relative to the next amino acid between the C alpha and C carbon, so they can actually twist and turn, but the planar structure is not going to change. They may twist and turn. Okay, the primary structure of protein is depicted here as beads on a string with the N terminus and the C terminus labeled. Um, the order in which you would read this peptide chain would begin with the N terminus as glycine, isoleucine, etc., and the end with methionine. So it ends with methionine but starts with glycine and isoleucine, right? So as you can see, these are the different beads on a string with N terminus and C terminus, right? So you can see that N terminus, C terminus. So it starts with lysine, ends with proline. Okay, so you can see the primary structure of protein, three-letter abbreviation indicates specific amino acid. There are 20 commonly found cells of living organisms. So lysine, valine, phenylalanine. Again, we have glycine, we have arginine, we have cysteine, we have leucine, we have alanine, we have methionine, we have arginine, lysine, histine, glycine, leucine. So all the different amino acids are connected in a bead. Okay. Secondary structure of proteins. The secondary structure of proteins comprises organized regions of polypeptide backbone stabilized by hydrogen bonds between atoms. All right. So secondary structure, what does it contain? It contains organized regions of polypeptide backbone stabilized by hydrogen bonds between atoms, right? So it has an organized region of polypeptide backbone and it has stabilized hydrogen bonds between atoms. Polypeptide backbone is composed of repeated CCN units where the side chain R is hanging on the first C. The second carbon is a carbonyl group and the nitrogen is amine NH group in amide. The polymer chain acquires conformation to achieve maximize hydrogen bonding between carbonyl and NH amine of the same chain, intramolecular or among the neighboring chain, and minimize the steric strain, the distortion of bond angles or distances caused by bulk of substituents around a central atom due to the R groups. This way, the polymer backbone acquires repetitive patterns in portions of the backbone. Right, so secondary structure of proteins comprise of organized regions of polypeptide backbone stabilized by hydrogen bonds between the atoms. The polypeptide bond is composed of repeated CCN units. The second carbon is the carbonyl group and the nitrogen is the amine. The polymer chain acquires conformation to achieve maximize hydrogen bonding. So you have to understand that the basic requirements of polymer chain acquires conformation to achieve maximize hydrogen bonding of the same chain or among the neighboring chain, intermolecular or intramolecular, and minimize steric strains, distortion of bond angles or distances caused by the bulk of substituents around the central atom due to its remaining groups. All right, so understand that eventually what secondary structure of protein is aiming at is to minimize the steric strain, the distortion of bond angles. This is why the polymer backbone acquires repetitive patterns in the portion of the backbone to minimize the steric chain. The two common secondary structure encountered in proteins are alpha helix and beta pleated sheet. Very important. Alpha and beta pleated sheets are questions that are usually asked on MCAT, so you will have to remember them. Other portion of polymer backbone that are regular but not repetitive are called random coils. Triple helix is another common secondary structure found in collagen proteins in connective tissue, right? So alpha helix and beta pleated sheets are very common. Other portion of polymer backbone that are regular but not repetitive are random coils. Triple helix is another common secondary structure found in collagen proteins in connective tissues. Basic secondary structure of proteins illustrate alpha helix, beta pleated sheet, random coil, and triple helix. So this is just a string, a ribbon structure, that's alpha helix. This is a beta pleated sheet As you can see, these are amino acid chains, and this is a pleated sheet. That's why it's called as beta pleated sheet. That this is not random coil, it's very irregular. This is a triple helix structure. As you can see, this is better designed, and it's a very sophisticated version, right? So this is the triple helix. Alpha helix. What is alpha helix? In the alpha helix portion, the polypeptide chain acquires a coil-shaped spiraling clockwise from N terminus to C terminus, right? Held by hydrogen bonds between the carbonyl groups and amine groups in the backbone, and side chain protruding outside, as illustrated in the model. Each amine is hydrogen bonded with carbonyl group 4 units away, shown by the hash lines in the model. Each side chain R group on an amino acid reside is protruding outwards from the helix. The alpha helix is represented by a right-handed spiral-shaped ribbon, as shown by the red color ribbon in the structure of glucagon. The side chain on each amino acid can be seen protruding out from the helix. A random coil portion of glucagon is shown by a blue color tube. Since the side chain hangs out in free space, short and long chain amino acid can easily be accommodated in an alpha helix without steric strain. Keratin, a fibrous protein of hair, fingernails, horns, and wool, is composed of major portion of alpha helix. Keratin, a fibrous protein of hair. So, keratin is a very important part of our hair, fingernails, horns, and wool. Is composed of major portion of alpha helix. That's exactly why, keratin bonds are actually broken when you try to straighten your hair, right? So, because of fibrous protein of its fibrous, so it's easy to heat it, but it also damages the proteins because when we heat anything, it kind of denatures the protein. So that's why it happens. All right. So alpha helix, remember, N terminus to C terminus held by hydrogen bonds, right? It is spiral shaped as a ribbon. The side chain on each amino acid can be seen protruding out of the helix. The random coil portion of glucagon is shown by blue color tube. Since the hang side chains hang out in free space. This is the secondary structure of glucagon. Alpha helix shown as red ribbon and random coil as blue tube. So these are just the random coils which goes, through the ribbon structure. Blue tubes with polypeptide backbone and side chain shown as stick model of the molecule. So all these are side chain, and this is the alpha helix red ribbon. So as you can see that, all right. So as you can see, there are protruding N and C terminus. Beta pleated sheet is a portion of polypeptide chain in which sections of polypeptide chain are aligned parallel or anti-parallel and held together by hydrogen bonds between carbonyl groups and the amine groups of neighboring polypeptide chains. The beta pleated sheet is usually represented as ribbon with an arrowhead pointed toward the N terminus. Since the polypeptide backbone is in a zigzag shape, multiple chains running parallel to each other in a beta pleated sheet is pleated shape. The two neighboring chains are parallel if both have N terminus on the same side. As shown on the right, the anti-parallel if the two adjacent chains have N terminus on the opposite side. The neighboring chain may be different polypeptides or the same polypeptide chain with a hair clip like bends at the ends. Let's take a look at it. The alternate carbonyl and amine on the backbone of the neighboring chain point towards each other and hydrogen bond with each other. Other set of alternate carbonyl and amine point to the other side, establish hydrogen bond with chains on the other side. Hydrogen bonds give mechanical strength to the proteins. Silk is made of fibrous protein called fibrin, with a significant portion as beta pleated sheet. The alternate side chain are point above and below in a beta pleated sheet. The amino acids in pleated sheet usually have short side chain such as glycine, valine, alanine, serine, as the long chain cause more steric strain. Alright, so as you can see, we can see the R remaining group. So these are the hydrogen bonds that are forming together, nitrogen and oxygen. We also see remainder group inside, and these are all anti-parallel. This is parallel. The other one is anti-parallel, right? So triple helix is a secondary structure found in collagen. Collagen is structural protein that is strong and elastic, present in connective tissue of the tendon, cartilage, blood vessels, skin, and bone. It is the most abundant protein of vertebrates, making 30 to 35% of proteins by weight. The collagen peptide is composed of repeated units of glycine XY, where X is usually proline and Y is generally hydroxyproline. Proline with a hydroxy OH group installed on the side chain. Proline has a secondary alpha amine group that causes bend in the peptide chain, which is needed for the helical structure of collagen peptides, right? So we learned in the amino acid lecture that proline has a proline is kinky, which has a kick. The reason why we said that is because it gives a bend in a peptide chain. The extra OH in the side chain of hydroxyproline allows more hydrogen bonding in the collagen peptide, which makes connective tissue stronger, right? So the extra OH in the side chain um, helps with extra hydrogen bonding in collagen peptide, which makes connective tissue stronger, right? So if we did not have an extra, then it would not collect to each other, right? And that's why we need uh, stability, and that's why we have an extra OH that is being used in collagen. Tertiary structure proteins, the three-dimensional arrangement of all the atoms of a single polypeptide chain in space held together by stabilizing interaction between the side chain and the backbone groups is called the tertiary structure protein. Three-dimensional arrangement of all the atoms of a single polypeptide chain in space held together by stabilizing interaction between the side chain, right? So if you have a three-dimensional arrangement of all the atoms of a single polypeptide chain in space, and it's held together by stabilizing interaction between the side chain and the backbone groups, is called the tertiary structure of proteins. Illustration of disulfide linkage, salt bridge, coordinate bonds with metal ions, hydrogen bonding, hydrophobic interaction that stabilize the tertiary structure of proteins, right? So as you can see, this particular is hydrophobic interaction. This is alpha helix. This is beta pleated sheet. These are salt bridges. This is again hydrophilic interaction, water loving interaction. This is hydrophobic interaction. These are just random coils. The blue coils are just random. This is a covalent bond, right? As you can see, the covalent disulfide bond, sulfur sulfur bond. This is hydrophilic interaction. One more time, this is an extra hydrogen. This is again a hydrogen bond, H3H3, right? Now this is a metal ion coordination. All right, so as you can see, this is a salt bridge. So we have disulfide linkage, we have salt bridge, we have coordinate bonds with metal ion, right? Hydrogen bonding and hydrophobic interactions stabilize the tertiary structure of proteins. So the tertiary structure of proteins have multiple interactions, right? Multiple bonding, multiple linkages, multiple interaction. It makes it stable because it has so many interaction that it can contain, right? So remember that tertiary structure is a very sophisticated form of primary and secondary structure. So what is disulfide linkage? When thiol groups of two cysteine residues come close to each other in a folded protein chain, they make disulfide linkage, the only covalent bond besides peptide bond holding amino acid residues together in proteins, right? So this is the disulfide linkage, the thiol group, right? So when thiol groups come together, groups of two cysteine residues come close to each other in a folded protein chain, they can make a disulfide linkage. Provided the protein chain is folded, two cysteine residues are coming together in a folded protein chain and make a disulfide linkage, right? The side chain of acidic amino acid asparagine loses protons and becomes anion. Whenever you lose something, you become an ion. And that of basic amino acids, lysine gains a proton, becomes a cation, right? So when you gain a proton, you become more positive, and that's why you become a cation because cats have paws, right? So you gain a proton, and you become positive. You lose a proton, you become anion. Okay. When the opposite ions come close to each other, they make an ionic bond, like that in salt, called salt bridge. CH2COONH3 plus CH24 can form between asparagine and lysine residues. This is an ionic bond holding opposite charges together. So when opposite ions come close to each other, they make an ionic bond like that in salts called salt bridge, uh, which includes CH2CONH3 plus CH24 can form between asparagine and lysine residues. This is an ionic bond holding opposite charges together. Some metals can act as Lewis acids and make coordinate covalent bond by accepting a lone pair of electrons from Lewis bases, right? Oxygen and nitrogen atoms in protein have lone pairs of electrons that can act as Lewis bases. All right. So, for example, one oxygen atoms of two carbohydrate uh, groups can make coordinate covalent bonds with magnesium Mg2 plus, resulting in coordinate covalent linkage, right? So between the two amino acids, right? So for example, an oxygen atom of two carbohydrate groups can make coordinate covalent bond with Mg2 plus, right? So they can create a coordinate bond between uh, two uh, elements and can create a covalent linkage. Hydrogen bonding. Hydrogen atoms bonded to oxygen or nitrogen atoms, example in OH and NH2, can make hydrogen bonds with any oxygen or nitrogen atom. These bonds could be between side chain, backbone groups, or side chain and backbone groups. For example, OH in the side chain of serine can make hydrogen with carbonyl oxygen of the amine group CONH2 of asparagine residue as shown below. So, as you can see, there's a hydrophobic interaction. Amino acids with aliphatic or aromatic hydrophobic side chain, valine, phenyl, and etc. establish hydrophobic interaction. Hydrophobic means water fearing interaction between valine and phenyl and any side chain shown in blue. Alright. So even if it's hydrophobic, it's still an interaction, right? So amino acid with aliphatic or aromatic hydrophobic side chain establish hydrophobic interaction. Hydrophobic interaction is London dispersion forces, an intermolecular force acting between atoms and molecules that are normally electrically symmetrical, that is, electrons are symmetrically distributed with respect to the nucleus. They are a part of van der Waals forces between nonpolar groups. The physiological medium is predominantly water. As a non-polar liquid like oil, when mixed with a polar liquid like water, the oil drops merge expelling water. Similarly, hydrophobic groups in proteins come close to each other due to hydrophobic interaction and tend to stay in interior of the protein, expelling water away from those regions. Ionic and polar compounds dissolve in water. Similarly, ionic and polar groups on the side chain stay on the protein's outer surface is interacting more with water. All right, so ionic and polar groups on the side chain stay on the proteins on the outer surface to interact with water. Hydrophobic groups of proteins come close to each other due to hydrophobic interaction and to stay interior of the proteins, expelling water away from those regions. Ionic and polar compounds, in the end, they dissolve in water. The tertiary structure of proteins may be predominantly alpha, alpha helix, beta, beta pleated sheet, or random coil, but often it is a mix of these. For example, carboxypeptidase A from bovine pancreas, tertiary structure carboxypeptidase A from bovine showing alpha helix in red, beta pleated sheets in yellow, and random coil is green. Alright, so this is like the tertiary structure where you have alpha, beta, and random coil coming together creating a big protein, right? So as you can see that. So what is the role of proline, right? The distinctive cyclic structure of proline side chain gives proline an exceptional conformational rigidity compared to other amino acids. It also affects the rate of peptide bond formation between proline and other amino acids. When proline is bound as an amide in a peptide bond, its nitrogen is not bound to any hydrogen, meaning it cannot act as a hydrogen bond donor, but a hydrogen bond acceptor, right? So when proline is bound as an amide in peptide bond, its nitrogen is not bound to any hydrogen. It accepts hydrogen as an acceptor. Peptide bond formation with incoming pro-tRNA in the ribosome is considerably slower than with any other tRNAs, which is general feature of an alkyl amino acid. Peptide bond formation is also slow between an incoming tRNA and a chain ending in proline, with the creation of proline-proline bond slowest of all. The exceptional conformational rigidity of proline affects the secondary structure of proteins near a proline residue, may account for proline's higher prevalence in the proteins of thermophilic organisms, right? So exceptional conformation rigidity, like we discussed before, the more stable a protein is, the better it can function. So the exceptional conformational rigidity of proline affects the secondary structure of proteins near a proline residue and may account for proline's higher prevalence in the proteins of thermophilic organisms. Why? Because if it has a conformational rigidity, what will happen? The process of denaturation will get reduced. So it doesn't matter if you're thermophilic, you're heat loving, but you're still not going to impact the bond. Why? Because proline has a side band which makes it more conformationally stable. Role of cysteine. Cysteine plays structural roles in proteins, can also participate in electron transfer reaction when some structural folds provide appropriated environments for stabilization of its sulfur hydryl group in an anionic form called thiolate Rs. In contrast, sulfur hydryl group of free cysteine has a relatively high pKa 8.5 and consequently is relatively inert for redox reaction in physiological condition. Thiolate is considerably more powerful as nucleophilic agent than its protonated form. Therefore, reactive cysteine are present mainly in its ionic form in proteins. Cysteine is very important. The reason why it's important is because of the sulfur hydryl group of free cysteine has a relatively high pK 8.5, right? So that's very important. Um, and it helps with physiological functions. All right, so what is the role of hydrophobic interaction? What happens? This hydrophobic interaction describes the gain in free energy on the transfer of nonpolar residues from an aqueous environment to the interior of the proteins, right? So nonpolar residues from an aqueous environment to the interior of the proteins. This has been accepted as one of the major forces involved in folding of proteins, responsible for affecting biology, including cell membrane, vesicle formation, protein folding, insertion of membrane proteins into the nonpolar lipid environment, and protein small molecule association. Hydrophobic interactions are important for the folding of proteins. This is important in keeping a protein stable, biologically active, because it allows the protein to decrease in surface and reduce the undesirable interactions with the water. Besides from proteins, there are many other biological substances that rely on hydrophobic interactions for its survival and function, like the phospholipid bilayer, like the phospholipid bilayer. Besides from proteins, there are many other biological substances that rely on hydrophobic interaction for its survival functions, like the phospholipid membranes in every cell of your body, right? So, besides from proteins, there are many other biological substances that rely on hydrophobic interactions. The hydrophobic interactions are very important for protein stability, especially for the interior as well as the outside, exterior. Um, Proteins on the cell membrane. Mostly it's interior because most of the hydrophobic interactions occur inside the cells. Okay, so the proteins that are residing inside the cells, they are more hydrophobic. So hydrophobic interactions are important for the folding of proteins. The association of more than one protein into closely packed arrangement is called quaternary structure proteins or quinary structure proteins. The individual protein in the assembly are called monomers or subunits. The subunits are held together by the same interaction that hold the tertiary structure of individual subunits. Hydrogen bonds, hydrophobic interactions, salt bridges, etc. An association of two protein is called dimer. So if there are association of two proteins, they are called as dimers. 3 protein is a trimer, 4 is a tetramer, 5 is a pentamer. All right. These subunits are structurally similar, about the same size, and work cooperatively in the hemoglobin tetramer. Conformational stability. The functional stability of proteins mostly depends on integrity of three-dimensional structure. The tertiary 3D structure of proteins results from folding and stabilization of the polypeptide chain. Higher structural and functional organization levels lead to quaternary. These supramolecular levels may result from non-covalent binding and or covalent bonding of homologous and or heterologous subunits, right? Covalent binding can be disulfide bridge between cysteine residues and other post-translation modification on amino acids, transglutaminase mediated isopeptide crosslinks, or on the glycan units in the case of glycoproteins. Denaturing. In biochemistry, denaturation is a process in which proteins or nucleic acid lose folded structure present in their native state due to various factors, including application of some external stress or compounds such as strong acid or base, a concentrated inorganic salt, an organic solvent, agitation, and radiation or heat. If proteins in a living cell are denatured, the results in disruption of cell activity and possibly cell death, right? So if proteins in a living cell are denatured, the result disruption of cell activity and possibly also cell death. Protein denaturation is also a consequence of cell death as well, right? So if a cell dies, it also causes protein denaturation. Denatured proteins can exhibit a wide range of characteristics from conformational change and loss of solubility or dissociation of cofactors, right? We discussed the cofactors are like synthetic groups attached, right? So cofactors to aggregation due to the exposure of hydrophobic groups. Loss of solubility because of denaturation is called coagulation, right? So we have a lot many processes that may have coagulation issues. Denatured proteins lose their 3D structure and therefore cannot function, right? So your the function of protein is fully dependent on 3D structure. So if the proteins are denatured, it may lose their structure, and function of any protein is defined by its shape, right? Its structure. The effects of temperature on enzyme activity. All right, increasing temperature increase the rate of reaction coefficient Q10, right? So whenever we increase the temperature, it will increase the rate of reaction coefficient. Now how fast the reaction is occurring, right? For example, we increase the temperature of water boiling, right? So we increase the temperature, it would increase the rate of reaction. uh, the middle graph shows the fraction of folded and functional enzyme decreases above its denaturation temperature. Right, so the fraction of folded and functional enzyme, it decreases above its denaturation temperature. Right, so this is the denaturation temperature. Now denaturation decreases activity, right, after it reaches a certain level of temperature, denaturation temperature. So there is a threshold of denaturation activity. Consequently, an enzyme's optimal rate of reaction is at an intermediate temperature. So this is the optimal rate of reaction. It is right in the middle. So temperature increases the activity. This is the denaturation decreases the activity. This is the optimal rate of reaction, and right here lies the intermediate temperature. Example of denaturing occurring in proteins. A classic example of denaturing is proteins come from an egg white, which are typically largely egg albumins in water. Fresh from the eggs, egg whites are transparent and liquid. Cooking the thermally unstable white turns them opaque, right? Forming an interconnected solid mass. The same transformation can be affected with a denaturing chemical. Pouring egg whites into a beaker of acetone will also turn eggs white, translucent and solid. The skin that forms on curdled milk is another common example of denatured protein, right? The protein albumin in the egg while undergoes denaturation and loss of solubility when the egg is cooked. Paper clips provide a visual analogy to help with conceptualization of denaturation process, right? So these are normal, but after you add the paper clips in an acetone, they start denaturing, they start opening up. So as you can see, they have kind of lost their conformational stability. So heating, has the same exact impact as pouring egg whites into acetone filled beaker, right? So we actually get the point. Anytime when you have chemical bonds breaking, it causes denaturation of protein, which also causes cell death, right? Okay, so functional proteins have four levels of structure organization, right? Primary structure, the linear structure of amino acids in the polypeptide chain. So primary structure is the linear structure of amino acids. Secondary structure has hydrogen bonds between peptide group chains in an alpha helix or beta sheets, right? So that's the secondary structure. Tertiary three-dimensional structure, alpha helix and a beta helix is folded. So tertiary structure is a higher form of sophistication from primary, which is just a linear structure of amino acid in the polypeptide. Secondary structure has hydrogen bonds. Tertiary has alpha helix and beta pleated sheet. Beta helix folded. Quaternary structure, three-dimensional structure of multiple polypeptides and how they fit together, right? So multiple structure, multiple polypeptides, way more sophisticated than primary, secondary and tertiary. Process of denaturation, functional protein showing a quaternary structure. When heat is applied, it alters the intermolecular bonds of the protein, unfolding of the polypeptide amino acids. The polypeptide amino acids are unfolded. How denaturation occurs at levels of protein structure. In quaternary structure, denaturation protein subunits are dissociated and or spatial arrangement of protein subunits is disrupted. So depending on the structure, how does denaturation occur? So in quaternary structure, how does it occur? Tertiary structure, how does it occur? Secondary, how does it occur? Primary, how does it occur? All right. So denaturation, protein subunits are disassociated and or the spatial arrangement of protein subunit is disrupted. Tertiary structure denaturation involves in the disruption of covalent interactions between amino acid side chains such as disulfide bridges between cysteine groups, non-covalent dipole-dipole interaction between polar amino acid and side chain surrounding solvent. Van der Waals induced dipole interaction between non-polar amino acid side chain. In secondary structure denaturation, proteins lose all regular repeating patterns such as alpha helices, beta pleated sheet, and adopt a random coil configuration. In secondary structure denaturation, proteins lose all regular repeating patterns such as alpha helixes and beta pleated sheets and adapt a random coil configuration. Primary structure such as sequence of amino acid held together by covalent peptide bonds is not disrupted by denaturation. Primary structure such as the sequence of amino acids held together by covalent peptide bonds is not disrupted, right? So primary structure is not disrupted by denaturation. However, quaternary, tertiary, and secondary will get disrupted, right? So in secondary structure, what happens? Proteins lose all regular repeating patterns such as alpha helixes and beta sheets and adopt a random coil configuration, right? Uh, so tertiary structure, how does it impact? The denaturation impacts a covalent interaction, then it impacts a non-covalent dipole-dipole interactions between polar amino acid and the surrounding solvent. And van der Waals are anyways very weak interactions between non-polar amino acid sites include dipole-dipole, right? So in van der Waals, it impacts, so each and every interaction of any kind will be disrupted, due to denaturation of tertiary. Protein folding consists of balance between substantial amount of weak intermolecular interactions within a protein. hydrophobic, electrostatic, and van der Waals interaction, protein solvent interaction, right? So protein folding consists of balance between a substantial amount of weak intramolecular interaction within a protein. So what does protein folding depend on? It depends on interaction within a protein. As a result, this process is highly reliant on environmental state that the protein resides in. This condition include and are not limited to temperature, salinity, pressure, and the solvents that happen to be involved, right? Consequently, an exposure of extreme stress, heat, or radiation, high inorganic salt concentration, strong acids and bases can disrupt a protein's interaction and inevitably lead to denaturation, right? So any exposure to extreme, say for example heat or radiation, when you heat your hair with a hot iron, what it does do, it kind of breaks the protein bonds in the keratin, right? So keratin is anyways fibrous, right? So it is very easy to denature keratin, and why? Because it happens due to heat. Solvation layer entropy. Solvation is critical for protein structural dynamics. Spectroscopic studies have indicated relationship between protein and solvent dynamics, and rates of gas binding to heme proteins in aqueous solution were previously observed to depend inversely on solution viscosity. The solvent-compatible enzyme Candida and tartic lipase B, which function aqueous organic solvents, was modeled using molecular dynamic stimulation. Data was obtained for the enzyme in acetonitrile, cyclohexane, N-butanol, and tert-butanol addition to water. Protein dynamics and solvation shell dynamics are characterized regionally for each alpha helix, beta sheet, and loop or connector region. The correlations are seen between solvent mobility and protein flexibility. So if the solvent is mobile, the protein will also be flexible. Solvation is critical for protein structural dynamics. It is seen that correlation is constantly higher when local solvent shell dynamics are considered rather than bulk viscosity. Protein flexibility is seen to correlate best with either the local interfacial viscosity or the ratio of mobility of an organic solvent in a regional solvation layer relative to hydration dynamics around the same region. So just remember one thing is that protein folding is very much dependent on its solvent, right? So remember that. Separation of one protein from all others is typically the most laborious aspect of protein purification. Separation steps usually exploit the difference in protein size, uh, physico-chemical properties, binding affinity, and biological activity. The pure result may be termed as protein isolate. So first is extraction, second is solubilization and stabilization, third is purification, and fourth is characterization and analysis. All right, so during the purification process, they will use chromatography, they will use centrifugation, they will use dialysis, they will also use electrophoresis, which we are going to learn. They may also use NMR, mass spectroscopy, circular and X-ray crystallography. All these things are very important because you will be asked questions on electrophoresis. You will be asked question on centrifugation, chromatography, mass spectroscopy, NMR spectra. All these are very important and you will be asked, trust me. Electrophoresis. What is electrophoresis? Electrophoresis separates proteins to a gel matrix based on size and charge by applying an electric field. One of the most common variants is SDS page, which uses a detergent SDS to denature proteins and separate them by size through polyacrylamide gel matrix. Electrophoresis is both a technique for protein separation and the analytical assessment of protein purity and molecular weight determination. Electrophoresis is the motion of charged dispersed particles or dissolved charged molecules relative to a fluid under the influence of a spatially uniform electric field. As a rule, these are Zwitter ions, right? So electrophoresis is the motion of charged dispersed particles or dissolved charged molecules relative to a fluid under the influence of spatially uniform electric field. All right. So electrophoresis separates proteins through a gel matrix based on size and the charge by applying electric field. So they apply an electric field, right? And because of that, it kind of separates proteins through a gel matrix based on the size. Electrophoresis is both a technique for protein separation and also for analytical assessment of pure protein purity and molecular weight determination. So, how pure is the protein? Electrophoresis is also a very important MCAT question. Trust me, you will be asked multiple questions based on it. Electrophoresis is used in lab to separate macromolecules based on their charges. The technique normally applies a negative charge called cathode. So protein molecules move towards a positive charge called anode. Therefore, electrophoresis of positively charged particles or molecules is sometimes called cataphoresis, while electrophoresis of negatively charged particles or molecules and ions is sometimes called anaphoresis. Electrophoresis is the basis for analytical techniques used in biochemistry for separating particles, molecules, or ions by size, charge, or binding affinity, either freely or through a supportive medium using a one directional flow of electric charge. It is used extensively in DNA, RNA, and protein analysis. Liquid droplet electrophoresis is significantly different from the classical particle electrophoresis because of droplet characteristics such as mobile surface charge and non-rigidity of the interface. Also, the liquid-liquid system, where there is an interplay between hydrodynamic and electrokinetic forces in both phases, adds to the complexity of electrophoretic motion. Agarose gel electrophoresis. Supporting media in this type of electrophoresis is agarose gel, which is very common. It is routinely used for electrophoresis of nucleic acid like DNA and RNA, right? So one is glass plate, which is like a casting tray. Second is a masking tape, pouring agarose solution onto the plate. Comb is pushed down into gel to form the wells, right? So uh, the comb, some a tool like this is kind of pushed down into the gel to form wells. Gel plate immersed in charged buffer solution. DNA segments loaded into wells with micropipette, right? So the DNA segments are loaded into wells with micropipette. Non-enzymatic protein function, transportation of molecules, structure, support, movement, immunity, nourishment. Non-enzymatic proteins are proteins that carry out functions that require the capacity to bind but do not necessarily catalyze a reaction like enzymes. Non-enzymatic proteins play an equally important role in our cells as enzymatic proteins. Many unique non-enzymatic proteins coexist to provide structure and signaling capabilities. Non-enzymatic proteins can also act as receptors, ion channels, transporters, motin proteins, and antibodies, right? So there are multiple non-enzymatic protein function. One is transportation of molecules, second is structural support, third is movement, also immunity, nourishment, and also capacity to bind. That is also one of the important function. Equal important role in our cells as enzymatic proteins. All right, so it not only has enzymatic function but also non-enzymatic functions, and you will be asked questions on what is protein binding? Protein binding describes the ability of proteins to form bonds with other substances and most commonly refers to the bonding of drugs to these molecules in blood plasma. Red blood cells are the components of blood and to tissue membranes. This binding is due to electrostatic and hydrophobic forces between drug and the protein. In the context of pharmacology, the amount of drug that is bound determines how effective it is in the body. The bound drug is kept in the bloodstream while the unbound component may be metabolized or excreted, making it the active part. So, if a drug is 95% bound to a binding protein and 5% is free, That means that 5% is active in the system and causing pharmacological effects, right? So let's say if a drug is 95% bound to a binding protein and 5% free, that means the 5% is active in the system and causing the pharmacological effects. All right, so the 95% is bounded to a protein, 5% is what that is helping actively in the system, causing its effects. One example is hemoglobin, the protein responsible for transporting oxygen from the lungs to the other parts of the body. It has four sides that can bind an oxygen molecule. The bond formed in this case is quite weak. It must be for the oxygen to easily remove where it's needed. In other cases, molecules may bind more strongly. The tendency of a drug to bind proteins and the strength of the bond are important factors in pharmacology. The proteins commonly involved in binding with drugs are albumin, lipoproteins, alpha 1 acid glycoprotein AGP. Binding is often reversible and in this case it can create a chemical equilibrium in which the chemical reaction can go backward and forward with no net charge in reactants and products. Okay, so protein plus drug, protein drug complex. So just remember that. If the concentration of unbound drug is reduced, some of the protein drug complex may split to release more of the compound, maintaining the equilibrium, right? Um, the the amount of binding and fraction of unbound written as the amount of unbound drug over the total amount are determined by the compound's affinity for the protein and their relative concentration. This is important when considering other medication that a patient might be taking. Certain proteins may already be saturated with which would affect the amount of free drug and possibly change the pharmacological effects. Role of proteins in enhancing immune system. So what is the role of protein enhancing immune system, right? We just said that non-enzymatic proteins have non-enzymatic function as well, and immunity is a part of it. The role of protein maintaining a strong immune system cannot be overstated, right? Proteins are essential nutrients that play a critical role in several essential processes within the body, including supporting the growth and repair of tissue, hormones, and immune function. Proteins are necessary to produce antibody, which are proteins that identify and neutralize harmful pathogens. Protein is also necessary for growth and maintenance of immune cells, such as T cells and B cells require protein to replicate and function properly. Without adequate protein intake, the body may not be able to produce enough immune cells to fight off infections and diseases, right? So it has multiple non-enzymatic function, helping the immune system become stronger. It includes supporting growth and repair of tissues, very important. Also helps with hormone production and immune functions. Proteins are also helping in production of antibodies, right? uh, and antibodies protect and neutralize harmful pathogens. Protein are also necessary for the growth maintenance of immune cells, such as T and B cells, very important. Require protein to replicate and function properly. Without adequate protein intake, the body may not be able to produce enough immune cells to fight off infection and diseases. Protein help reduce inflammation, right, which is very important for immune system response to infection and injuries. Inflammation is a natural response to harmful stimuli, but chronic inflammation can lead to serious health problems. Protein can help reduce inflammation by promoting production of anti-inflammatory cytokines and reducing the production of pro-inflammatory cytokines. Arginine stimulates the immune system and promotes wound healing. It plays a vital role in production of T cells, a type of immune cell that recognize and destroy specific pathogens. Cysteine is necessary to produce glutathione, a powerful antioxidant that helps protect immune cells from damage caused by free radicals. Methionine is another amino acid that is important for immune system function as it is required for the synthesis of proteins that are involved in immune cell signaling and also in communication. What are motor proteins? Motor proteins are a class of molecular motors that can move along the cytoskeleton of the cells, right? They convert chemical energy into mechanical work by hydrolysis of ATP, adenosine triphosphate. Flagellar rotation, however, is powered by a proton pump, right? So kinesin walking on microtubule using protein dynamics on nanoscales, right? So motor proteins are a class of molecular motors that can move along the cytoskeleton of the cells. Motor proteins are the driving force behind most active transport proteins and vesicles in the cytoplasm. Kinesins and cytoplasmic dyneins play essential role. Dyneins is also one of the protein. Intracellular transport such as axonal transport, transport axons, and in the formation of spindle apparatus and the separation of chromosome during mitosis and meiosis, right? So symbol apparatus. Axonal dynein found in cilia flagella is crucial to cell motility, for example, in spermatozoa, and fluid transport, for example, in trachea, right? So, axonal dynein found in cilia and flagella, very important, motor protein, dynein, and kinesin, responsible for flagellar movement in spermatozoa. So sperms, the flagella, the backside, that the tail that wiggles is because of dynein. The muscle protein myosin motors the contraction of muscle fibers in the animals. So muscle protein myosin motors the contraction of the muscle fibers in animals. Yes. So myosin is responsible. The motor protein myosin is responsible for muscle fiber contraction in all the animals, which includes us. Diseases associated with motor protein defects, right? So very important, the clinical application. The importance of motor proteins in cell becomes evident when they fail to fulfill their function. For example, kinesin deficiency have been identified as Charcot-Marie-Tooth disease and some kidney diseases. Dynein deficiency can lead to chronic infection of the respiratory tract as cilia fail to function without dynein. Numerous myosin deficiencies are related to disease states and genetic syndromes because myosin 2 is essential for muscle contraction. Defects in muscular myosin predictably causes myopathies. Myosin is necessary in process of hearing because of its role in the growth of stereochelia. So defects in myosin protein structure can lead to Usher syndrome and non-syndromic deafness. Right, so myosin deficiency are related to disease states and genetic syndromes. Cytoskeleton motor proteins. Motor protein utilizing the cytoskeleton for movement fall into two categories based on their substrate, microfilaments and microtubules. So, cytoskeleton motor proteins utilize cytoskeleton for movement, and they usually fall into two categories. One is microfilament, and another is microtubule. Actin motors such as myosin move along microfilament through interaction with actin, and microtubule motors such as dynein and kinesin move along microtubules through interaction with tubulin. There are two basic types of microtubule motors, plus end motors and minus end motors, depending on the direction in which they walk along the microtubule cables within the cell. Actin motors, myosin. Myosins are superfamily of actin motor proteins that convert chemical energy in the form of ATP to mechanical energy, thus generating force and movement. The first identified myosin 2, is responsible for generating muscle contraction. Myosin 2 is an elongated protein that is formed from two heavy chains with motor heads and two light chains. Each myosin head contains actin and ATP binding site. The myosin heads bind and hydrolyze ATP, which provides energy to walk towards the plus end of an active filament. Myosin 2 Myosin 2 are also vital in the process of cell division. Right? So actin motors, myosin is very important because it belongs to the superfamily of actin motor proteins which are responsible for converting chemical energy to form ATP. Myosin 2 is responsible for generating muscle contraction in animals we just studied. Myosin 2 is an elongated protein that is formed from two heavy chains of motor heads and two light chains. Each myosin head contains actin and ATP binding site. Myosin heads bind with hydrolyzed ATP, which provides energy to walk towards the plus end of an actin filament. Microtubules. All right. So what is a microtubule? A superfamily of related motor proteins that use a microtubule track in an enterograde movement, vital to spindle formation and mitotic and meiotic chromosome separation during cell division, and are also responsible for shuttling mitochondria, Golgi bodies, and vesicles within eukaryotic cells. Kinesins have two heavy chains and two light chains per active motor. The two globular head motor domains in heavy chains can convert the chemical energy of ATP hydrolysis into mechanical work to move along microtubules. The direction in which cargo is transported can be towards plus end or the minus end depending on the type of kinesin. The kinesins with N-terminal motor domains move their cargo towards the plus end of microtubules located at the cell's periphery, while kinesin with C-terminal motor domains move cargo towards the minus ends of microtubules located at the nucleus. 14 distinct kinesin families are known, with some additional kinesin-like proteins that cannot be classified into these families, right? So microtubule motor kinesin is also very important. It is a superfamily of related motor proteins, vital to spindle formation during mitosis and meiosis. Kinesins have two heavy chains and two light chain per active motor. Two globular head motor domains in heavy chain can convert chemical energy of ATP hydrolysis into mechanical work to move along microtubules. So microtubule motor dynein is also very important protein. Microtubule motors capable of retrograde sliding movement, the backward movement. The complexes are much larger, more complex than kinesin and myosin motors, composed of two or three heavy chains and a large variable number of associated light chain. They drive intracellular transport toward the minus end of the microtubules, which lies in the microtubule organizing center near the nucleus. The dynein family has two major branches. Axonal dyneins facilitate the beating of cilia and flagella by rapid and efficient sliding movements of microtubules. Another branch is cytoplasmic dyneins, which facilitate the transport of intracellular cargoes. Compared to 15 types of axonal dynein, only two cytoplasmic forms are known. All right, so thank you for watching. This was introduction to protein. I hope it helped. We have covered all the points that you need to learn about primary, tertiary, secondary, tertiary structure of proteins. What is folding, binding, conformation, stability, what kind of motor proteins are asked in AMC, and we've seen that as well, and also the function of protein itself. If you have any questions, please ask. Alpha and beta sheets are very important. Electrophoresis is very important. Chromatography is very important. Spectroscopy is very important. MRI is very important. X-ray crystallography is also very important. So certain topics are usually asked in most of the MCAT exams, and I insist that you listen to it, you read about it, because it will be asked, and I promise you, that it will be asked. All right. So thank you very much for watching introduction to proteins. I will come back with more videos. After this, I'll be talking about enzymatic functions and have few more videos coming in. So please come back, like, share, subscribe, and comment. Bye.